two of our invited speakers. The first of those is Robb de Vries from the University of Utrecht. Rob did his PhD in the Faculty of Veterinary Medicine at Utrecht University. He then moved to the Scripps Research Institute for postdoctoral work, which is awarded a Rubicon grant. And then in 2014, he returned to the Netherlands, joining the Department of Chemical Biology and Drug Discovery, where he's now a principal investigator. I'm sure he'll be known to many of you for the really exciting work he's doing on glycan binding, for which, amongst other things, he was recently awarded an ESWI Best Body of Science award. And he's going to be talking today on receptor binding specificity of influenza A virus. So, Rob, thank you very much.
Yeah, Ed, thank you for the introduction, and also thank you for inviting me for this workshop. It's my pleasure. So can you see my screen and my mouse? Yeah. Okay, great. So just start. So I think that understanding influenza virus receptor specificity is eventually essential for vaccine development. And I always start with this picture here, because literally every cell is covered with this dark matter of glycans, and it's something we don't understand much about, and hence the dark matter. But we do know that there are a lot of different ending glycans, and I was glad I was able to touch upon them a little bit earlier. And one of the interesting things that I would like to mention is that these N-glycans in nature are hardly symmetrical, as depicted here, because we are looking at an asymmetrical glycan. So we have these N-glycans on the cell surface, and we know that flu binds on the cell surface. And we like to think about these N-glycans in like trees. Actually, my favorite is kelp, because I think it's a little bit more fluidic than a tree. And you can start at the bottom with the glycoprotein, with the the bottom part, with what kind of glycan you're looking at, with different branches going into different arms, and eventually into the leaves, which are different sialic acids. And these sialic acids can contain a lot of different modifications as well. And in this presentation, I will go a little bit through this tree to demonstrate how all these different features are actually important for influenza A virus receptor binding. And as mentioned before, sialic acid linkage specificity is a host barrier, which also makes sense if we look at where these different sialic acids are expressed. So this is a lectin stain with plant lectins, so it's not super correct. But we, for example, know that human type receptors, alpha-2,6 linked sialic acids that human viruses bind, is that we express that predominantly in our upper respiratory tract. So it makes sense that if you sneeze, you spread this virus very efficiently. Interestingly enough, ferrets, our favorite animal model, have a similar receptor distribution in their respiratory tract. Also a lot of alpha-2,6 linked sialic acid in their nose, and they sneeze. Pigs, on the other hand, they also express a similar sialic acid linkage distribution, but the majority of them are actually modified in N-glycolil, and I will touch upon that a little bit later. Whereas in avian species, we see predominantly alpha-2,3 linked sialic acids in their respiratory tract and also in the gastrointestinal tract, whereas a lot of these viruses in avian species have a different route of transmission. So if you look at sialic acid linkage, as John already mentioned, you have two different linkages, whereas avian viruses prefer the sialic acid if it's linked to the third carbon of the penultimate galactose, whereas human viruses prefer the sialic acids if it's linked to the sixth carbon of the penultimate galactose. And this penultimate galactose is almost always linked, or influenza viruses prefer binding to these structures if there's an acetylglucosamine. And in the rest of my presentation, I will show these different symbols depicting sialic acids and in the yellow circles, galactose, and a glucnac in these blue squares. And just to make it a little bit more clear, if we have an avian type receptor, an alpha-2,3 linked sialic acid, the rest of the glycan points out of the receptor binding site, whereas the linkage difference makes a different structure of these glycans. So with this different 2,6 linked sialic acid, you can now have these glucanacs and gals and the other LACNAC structures, because we call this disaccharide a LACNAC, which goes over other structural features of HA. And this is the receptor binding specificity, whereas indeed sialic acid lies in the receptor binding pocket in all these different subtypes in the same way. And if we look at what kind of N-glycans are present in our respiratory tract, and this is a study from almost a decade ago where they actually grind up lung tissue, so we don't know where these N-glycans are. But interestingly, what we do see is that you have these very complex N-glycans with multiple LACNAC repeats. And that was of interest to us because we wanted to make these structures because they're present in the human respiratory tract. And in the last couple of years, a PhD student in my lab started to make, instead of these symmetrical glycans, because here with mass spec, we know the mass of the N-glycan, but we don't know the features. We don't know if all these 6 LACNAC structures are on one arm or are evenly distributed in both arms. So we wanted to make asymmetrical glycans, because making symmetrical glycans is pretty straightforward. So we start with egg yolk. When I was in the Boston lab, we literally cracked open eggs. Now you buy the powder at Sigma. And when you purify this glycopeptides, you can get it in very high purity and in massive amounts. You get this alpha-2,6 linked single LACNAC N-glycan. And then we start modifying it in the lab. And we do this all by enzymes. There's hardly any chemistry involved anymore. And I'm not going to dwell on all the different steps, because we know the availability, what John just mentioned, of these N-glycans is very limited. And we don't make these N-glycans in multi-milligram scales. Because for glycan arrays, we only need just 0.5 milligram in total or something. But we can desymmetrize these N-glycans by enzymes, because we know that the galactosidase from E. coli really prefers this alpha-3 arm, and so you end up with an asymmetrical N-glycan. And on the other hand, ST6-gal1, human ST6-gal1, prefers this lower arm to put a sialic acid, which forms a capping structure there. And if you don't push the reaction, you can purify these structures, and from that you can enzymatically modify these N-glycans at will. You can start modifying the lower branch, remove sialic acids, add 2,3 linked sialic acids, or add another 2,6, etc., etc. So we can actually make these N-glycans now asymmetrically at will. And for example, here on the right-hand side, we can elongate the alpha-2,6 arm also at will. So we can make all these different N-glycans. And previously, we already have shown that making these N-glycans chemically, we see differences in binding specificity of these different viruses. So when we're done, we print these glycans on glass. And this is a little movie with a non-contact printer where you add your 96 well plate, you get a pin, goes into that, and it will deposit defined N-glycans at a specific spot. And if you then have a glycan array, you can interrogate all these different glycans that we just made in a single assay. And some glycan arrays have up to 500 structures. We are now at around 200 different glycan structures. And you have a couple of ways to assess these glycan arrays. But again, all these approaches are based on multivalency. So you can immediately take the virus and add them on the array and detect it with serum or antibodies. But most of these viruses are BSL-2 or up, and the glycan array printer and reader are mostly not in an ML2 facility. So what we and others like to do is express these trimeric hemagglutinins, and before we can interrogate an array, we have to make them multivalent. And that's what we do here with a trimeric HA, and we use an antibody against the tag, which can be his, in our case, a strep. And then we add another antibody, against the mouse antibody or the human antibody to make a pre-complex. So you have 4 HA trimers here, and you can play around with the different labels on these antibodies. And then you can analyze which kind of glycans this HA or virus binds on a glycan array. And recently, and I want to touch upon this a little bit, we started to make fluorescent HAs. So while expressing it on the C terminus, we add a superfolded GFP. We now add also M-orange or plum to look at different wavelengths. And if we added this on the C terminus, actually at the time, Ilhan, as an undergraduate, was making these proteins, and he put this figure together in paint. And he came to my office and he was there like, well, this is how it looks. I said, well, let's see. We sent these proteins to Andrew Ward at Scripps for negative stain analysis. They were neighbors when I was, lab neighbors when I was there. And indeed, you have a trimeric HA with this trimerization domain. It was actually the first time that we saw this trimerization domain. And we have 3 superfolded GFPs hanging around on the C terminus. We now can also make them on the N terminus and even both. Then we have 6 fluorescent probes. And funny enough, if we then add these proteins by themselves, so no pre-complexation, just a single HA trimer, human type specific proteins bind on the array. They bind to complex N glycans, whereas avian HAs don't. But when we put these proteins on tissues or on cells, they both bind. So for that experiment, which was published last year in Journal of Molecular Biology, we know that avian HAs cannot bind by themselves on a glycan array, but they need a complex presentation of N glycans or glycans on the cell surface, enabling multivalency from the other side. So you can make HA trimers multivalent. If you have an HA glycan array platform, it's not always presenting everything the correct way, but on a cell surface it does. So that was one of the features that I wanted to touch upon, that a single HA trimer, a human HA trimer, can engage a complex N glycan to bind human type receptors. Other things that we do before I go into the whole H3 and 2 story, and like I said, I wanted to go a little bit through the tree, is that we recently also made these N glycans with different sialic acid linkages, but also with a modification at the C5 position. So whereas in humans, we have this N-acetyl structure, because we lack the enzyme that converts the CMP sialic acid to an N-glycolyl form. We were interested in how these equine and swine viruses would bind this N-glycolyl or not. And other species also actually lost this enzyme that converts the structure, for example, canine species and BCK cells. And then you can read more about this in the paper that appeared last year in Cell Reports about these different specificities to N-acetyl and N-glycolyl. Finally, before going to H3, we also analyzed different Lewis X specificities. So these LACNAC chains, and we made them linear structures here, you can modify them with different fucose structures. And normally when people do this, they make these structures enzymatically, and every GLUCNAC is fucosylated. But while we were synthesizing this, we actually added protecting groups on the N-acetylglucosamines to avoid putting fucose structures everywhere. And we were able to make all these different fucosylated LACNAC structures to assess if this would, get different specificities of avian HAs. Importantly, you can never have an alpha-2-6 linked sialic acid here if you have a fucose, because these enzymes are mutually exclusive and are not present in nature. So we didn't want to make that. And for example, here, we know, especially for H5N6 viruses, was touched upon earlier, that some of them have a preference for terminal Cia-Lewis X, whereas other viruses, or the prototypical Vietnam strain, will not bind the Cia-Lewis X. And during our studies, we were able to pinpoint these specificities to 2 amino acids in the 220 loop. where you can convert Cy-Lewis X specificity or non-modified LECNEC structures. And then we are now in progress to make these kind of structures also on N-glycans. So the big thing here in this workshop is antigenic drifts. And I always think that H3N2 evolution is outsmarting us because of these drifts. And here on the left, we have this phylogenetic tree, and all the different colors are different vaccine strains, because these viruses escape antibody-mediated immunity, and we need to update our vaccine strains every couple of years. And I always like to mention that it stops here at 2002, because I think after that moment, we have no clue anymore what's actually happening. And correct me if I'm wrong. So this virus is now present in the population for half a century. And what we see is that these viruses have a declining receptor binding avidity. They hardly bind to erythrocytes anymore. And also these viruses grow very poorly on MDCK cells, the very contemporary viruses. And one of our hypotheses is that we just have a very poor understanding of receptor specificity of these contemporary H3N2 viruses. And one of the, I guess the preferred, or one of the assays that everybody who works on influenza viruses likes, is the hemagglutination assay. So if you have red blood cells and you don't do anything, they will fall in a drop. But if you add a virus or these proteins pre-complexed, you will see hemagglutination. And with antibody serum test, you can inhibit that and you get an hemagglutination inhibition test. And I think this test is preferentially used in the laboratory to analyze antigenic drift and see the difference in hemagglutination inhibition by diluting the serum. And I started to work on this while I was in the Boston group, and there we already had, we started with analyzing different H3 proteins. These are proteins from 1986, 75, 89, 03, 09, 2011. This was originally submitted in 2014. And we can indeed see that these H3N2 viruses have an increasing amount of N-glycosylation all over the head of the HA. And when we do an ELISA, and here we have linear structures coupled to polyacrylamide, which polyacrylamide can bind on plastic, so you can actually do an ELISA. We noticed that after 2003, we don't get any signal in the ELISA anymore. And one of the features here, and I would like to draw your attention on structures 120 to 135. All these viruses bind alpha-2,6 linked sialic acids. These are all different linear glycans, O-linked glycans, and N-linked glycans. And on one moment, I hope you can appreciate it, is that we see this calm-like structure. Some glycans are not bound anymore by more contemporary H3N2 viruses. And to put this in a nutshell, for example, glycan 118 is not bound, only has 2 LECNEC structures, whereas 120, 121, 122, which has at least 3 LECNEC structures on an arm, are bound by these viruses. It doesn't really matter what kind of N-glycan core you have it on, because if you have 3 arms, you still need 3 LECNEC repeats. Core fucosylation doesn't really matter. If it has 2 LECNEC structures, these viruses, contemporary viruses, don't bind these structures, but do bind structures which have 3 LECNEC repeats. And here, as you may notice, these glycans are all symmetrical. Well, they're not exactly symmetrical because they're not from nature, but they're synthesized symmetrically, right? They all have the same kind of arm length. So the next step that we proposed at the time is that human type receptor specificity is a bidentate binding event, and not just for H3N2, because if we mutated H7, H10, H5 hemagglutinins to make them specific for human type receptors, we saw that they always preferred these N-glycans if they have 3 LECNEC repeats on an arm. And even in, for example, H7, with a single amino acid in the 190 helix, we were able to put this specificity on and off. Because if there's a lysine here, which is present in these strains, and if we convert it into a threonine, these more simpler amino acids, which you would see in H2 or human H3s, now we saw binding to these structures on the array. And the hypothesis at the time was that if you have this structure that is big enough, that you form a bidentate binding process with 2 sialic acids of a single N-glycan in 2 receptor binding domains within this HA trimer. So recently we started to work on this again, and we started with an hemagglutination test of a couple of different viruses, 91, 2003, 2009, 2017, 19, etc. And we added these viruses, and this work now, actually Fred here got some help from Rosanne, and we deposited this study recently on bioarchives. And if we add these viruses to chicken, turkey, and guinea pig erythrocytes, because guinea pig erythrocytes are currently used for hemagglutination inhibition assays. And what we see here is that after 2009, these viruses of the 3C2 clades don't bind any erythrocytes anymore. And I guess there are examples that bind guinea pig erythrocytes, but still, if they bind, and when we see it, the titer is really low. And interestingly enough, these contemporary 3C3 viruses, there are 2 antigenic clades now, regained binding to turkey and guinea pig erythrocytes. But this gap, that was our interest. Why do these contemporary viruses do not bind any erythrocyte anymore? And we actually added these viruses directly on the array. And to avoid serum or antibody detection, we actually use stem antibodies to detect the virus on the array. So we could just use one antibody to detect all the viruses. And in the 91 virus, we actually saw that it bound to very simple N-glycans. It only needed 1 LAC-NAC repeat, as we've seen before. In 2003, binding to this very simple N-glycan is lost, but still able to bind some N-glycans that only have 2 LAC-NAC repeats, and this is the whole array that we present here. In 2009, we can actually already see that glycan 13 is not bound anymore. Still some binding to glycan 10, 2 LAC-NAC repeats on both arms, but glycan structures 14, 15, and 17 are preferentially bound. And that's what we also saw for the viruses from 2017 and 2019. And what these N-glycans have in common is that they have a single arm with at least 3 LAC-NAC repeats. If we made these asymmetrical N-glycan and we removed, or we didn't extend the other arm, we didn't saw a big difference. So we thought like, oh, so maybe the bidentate binding is not true anymore if it's on a membrane. We also tested the 3C3 viruses, and again, 91, 03, and 09, you see an increasing loss of binding very simpler N-glycans. And this predecessor of the 3C3 antigenic clade actually still have the same receptor binding specificities we saw for contemporary viruses, but after that they regained binding to erythrocytes, as we've seen before, but we also see that these viruses now bind to more simpler glycan structures with at least 2 LAC-NAC repeats on their arms. So we saw this different receptor binding evolution profiles of these viruses. Then we wondered, okay, what kind of N-glycans are actually present on these different erythrocytes? So by releasing the N-glycans from erythrocytes, from both chicken or turkey erythrocytes, and we plotted these N-glycans by the presence of the amount of LAC-NAC repeats. For chicken erythrocytes, we hardly saw any N-glycans that have more than 3 LAC-NAC repeats. So they might have an N-glycan, a 3 LAC-NAC arm on one arm, but that's very unlikely. Whereas for turkey erythrocytes, we see indeed an increase in the amount of LAC-NAC repeats. And here you have a couple of N-glycans that hypothetically can have an arm with 3 LAC-NAC repeats terminating in a human type receptor. But we wanted to control this. So we did the same assay as we make these N-glycans. And actually, this is a very old assay because I think a lot of people can remember recellular a red blood cell. So you start with a red blood cell, you remove the sialic acids, and instead of adding an 2,6 or 2,3 linked sialic acid directly, we added an additional step. So while we were cleaving off sialic acids, we were at the same time, this is a one-part assay, extending these arms with beta-3-GNT2, a gluconate transferase, and B4-GalT1, which is a galactose transferase, to increase the length of the arms of these erythrocytes. And thereafter, we would recellulate these erythrocytes again. So we first tested the erythrocytes, what kind of N-glycans we actually put on. And indeed, for chicken erythrocytes, we are now able to make N-glycans that have 4 LACNAC repeats on their arms. So that's an increase. And also for turkey erythrocytes, we also saw an increase of N-glycans of relative abundancy of these glycans with longer arms. But actually, we were a little bit disappointed because we hoped that we would make a massive amounts of N-glycan with increasing arm length. But in the meantime, we were testing these erythrocytes. So on the x-axis, we have unmodified erythrocytes, just recellulated erythrocytes, non-recellulated erythrocytes, but do have long arms because polylacnacs. And then we have our erythrocytes that we just meant that we have here that have polylacnac structures and are recellulated with alpha-2-6-linked sialic acid. So for the 91 virus, we don't expect a big difference because the wild-type virus can hemagglutinate unmodified erythrocytes. We see for the 2003 virus, it's not able to bind these unmodified erythrocytes from chicken, because we did it both on chicken and turkey erythrocytes. But if we recellulate it with 2,6, we do see hemagglutination. For 2009, here comes the big difference, right? It only will bind recellulated turkey erythrocytes, but these 2017 and 2019 viruses that only bind these very complex N-glycans on the array, they only bind these erythrocytes with polylacnac repeats on their arms, recellulated with 2,6. Also the case for the older 3C3A virus, but because they readapt to shorter N-glycans, we can also see that they bind unmodified or just recellulated turkey erythrocytes. And I would like to draw your attention on the fact that our glycan-modified erythrocytes are always bound at the highest titers by these viruses, regardless of their from the year of isolation. So the next step, what we wanted to know, we really want to put our erythrocytes to the test. So we took a large panel of H3 and 2 viruses, ranging from 1968 until a year ago, a little bit more than a year ago, year and a half more or less. And we analyzed them on unmodified erythrocytes, recellulated erythrocytes of both chicken and turkey origin, and guinea pig erythrocytes. So what you can already see is that these viruses, because they're ordered from 68 to contemporary viruses, they hardly bind unmodified erythrocytes. They become negative pretty soon. If you recellulate them, you will get some higher titers. But also very quickly in evolution in the early 2000s, they will not bind these erythrocytes anymore. Very similar picture for the turkey erythrocytes, but we lose hemagglutination completely a little bit later. But now when we have our glycan-modified erythrocytes, so with long arms and alpha-2-6, we can see that all these viruses now hemagglutinate these erythrocytes again. So we now made erythrocytes that all H3 and 2 viruses bind, except for a couple of very heavily adapted vaccine strains. So the next step was to do hemagglutination inhibitions with these glycan-modified erythrocytes. That's what we did. So we have a couple of viruses that were in circulation at the time with their corresponding vaccine strain, post-infection ferret sera, which we got from Erasmus University in Rotterdam. And we can see that the homologous titers using these erythrocytes are very high. And we can confirm that the 3C2s and the 3C3s are antigenically very distant from each other because these sera do not provide any cross reaction. And I would like to draw your attention a little bit to the Kansas strain, which was one of our latest vaccine strains. It was actually only protective against the homologous virus. Yeah, we actually did the exact same experiment in the focus reduction assay. So where you use serum dilutions for infection of MDCK cells, exact same sera, exact same viruses, and we compared the focus reduction titers with our HA titers. I didn't draw an exact line through it, but I hope you can appreciate that the correlation is very high. So we can now do hemagglutination inhibition assays for contemporary H3 and 2 viruses again. So with that, I would like to summarize. I showed you how we chemoenzymatically synthesize N-glycans and do our own array development, not just for human viruses, but also for a large panel of avian viruses to look at different sialic acid linkages, modifications, arm modification. That's what I showed you with influenza virus receptor specificities. And I showed you that we can now do antigenic analyses of these H3 and 2 viruses by hemagglutination inhibition. The 2 PhD students actually are setting up, just actually set up a company to commercialize these erythrocytes. But to start up, I mean, we are very willing to send them out. We can keep them for several months after glutarate fixation for people to test these erythrocytes. So please don't be please ask if you want to have some erythrocytes. And with that, I would like to thank the people in my lab, especially Fred and Rosanna. Fred is a chemist who made all these different glycans, and Rosanna helped them out with all the hemagglutination assays. I showed you the work of Ilhan. I'm in the department with Professor Geert-Jan Boons. I would like to thank my collaborators at Erasmus, of course at Scripps Research, with whom a lot of this work was not done. The models that I showed you, actually I forgot to add their names there, with the bidentate binding event, are made by Oliver Grant and Robert Woods from the Complex Carbohydrate Research Center at the University of Georgia. And I would like to thank other collaborators at Mount Sinai, Amsterdam Medical Center, and Hokkaido University. And I'm continuously funded now by the NWO, which was recently, a week ago, hacked. So all our funding agencies is like totally put out now by the ERC and by the Royal Dutch Academy of Arts and Sciences. And with that, I would like to thank you for your attention.